Nano is basically 10 to the power minus 9. And when you refer to nanomaterial, it is 1 into 10 to the power minus 9 to 100 into 10 to the power minus 9 range. So 1 to 100 nanometers is what you're looking for when you talk of nanometer range. So here you are. You should remember ideally what is the size of some of these. For example, if you know water would not be in nanometer range because it is 0.1 nanometer. Glucose will be in nanometer range. Antibody would be in nanometer range. A virus would be just about the nanometer range. Cancer cells are definitely not in the nano range, right? So nano range is 1 to 100 nanometers. So you must remember ideally that this is your nano scale we're talking of, 1 to 100 nanometers, right? So then, of course, we move on. Historical perspective. I've told you there is this book called Engines of Creation by Eric Drexler. This book came in 1985, and it was a very important path-making book in the sense that this brought nanotechnology as a term into the common conscience of people, and people started getting to know that there is something called nanotechnology, where you can make machines so small that they can be used for, they can have properties very different from normal machines and can be used for very novel functions. And then, of course, the term was given by Norio Taniguchi. This also you must remember. He's a Japanese scientist, 1974, Norio Taniguchi. And the initial thought of nanotechnology came from the Richard Feynman lectures, where he gave a famous lecture known as "There is Plenty of Room at the Bottom" at Caltech, California Institute of Technology. And that is where the idea of nanotechnology began, right? And in this lecture, he also said that, at the nanoscale, it will not be gravity that would be the most important. It would be the van der Waals forces or the interaction forces that are there between two molecules that would be most important, right? Then, of course, Eric Drexler, the coming era of nanotechnology, you know that. And then, of course, the initial nanomaterial used to be in the form of fullerene. It is an allotrope of carbon. If you see, it is of a type of a football. And then, of course, you had what is known as graphene. And this is what led to the formation of nanotubes. Graphene is basically one layer of graphite, right? So graphene, isolated atomic layers of graphite, which is a flat mesh of regular hexagonal rings, can be seen as an extreme example of the family, and it is used to make carbon nanotubes. Then, of course, nanobiotechnology is the interface of nanotechnology and biotechnology. It includes applications of nanotechnology in the life sciences. Nanobiotechnology is the application of nanotechnologies in biological fields. Anything that uses a biological starting material, a biological design principle, because cells are regularly working at nanoscale. Most of your proteins are in nanoscale. Most of your antibodies, everything are in nanoscale. Your DNA itself is a nanoscale, so it has a 2 nanometer diameter or 20 angstrom diameter. RNA also are in the nanoscale. So therefore, the cell has been working at nanoscale since the time it arose, and now only we are starting to learn that at nanoscale things become different. So many of these design principles are coming from the actual biological experiences or experiments that we make. Then, of course, it has a biological or biomedical application, and it is using something at a nanoscale is what will be your biosensor or nanobiosensor. Integration of nanomaterials with biology has led to the development of diagnostic devices, contrast agents, quantum dots you've talked of, right? Diagnostic devices, we talked of Nanopore, if you remember, or MinION, the machine that is used for sequencing. We'll get to that here. And of course, analytical tools, therapy, we have talked of nanoparticles in cancer again. Drug delivery vehicles, we talked again of nanoparticles and how you can add both a contrast agent and a drug together to make what is known as a theranostic particle, right? So nanobiotechnology is rapidly evolving field, particularly with respect to biomedical applications, and therefore it is important. We talked a bit about this, nanobodies. These are very small antibodies, much smaller in size than your standard antibodies, but then they have still the same specificity. And then, of course, nanobiotechnology is being used in all areas of science that you can think of, for biological sciences specifically, in diagnosis, in gene therapy, in tissue engineering, in drug delivery, in pathogen detection, and food safety. And in Unit 2, when we talk of biosensors, we'll talk more about this in Unit 2. Then, of course, you remember we talked about how it is being used everywhere. If you talk in terms of crop improvement, so right from monitoring the soil to monitoring the weather to controlling how much moisture it should have, controlling a drip irrigation, Then of course, with the smartphones and smart technologies, you are directly connected with a chip, nano chip, to the plant, and you can monitor everything. So all those kind of things can be done using, the nanobiotechnology systems or nanobiosensors. And then, of course, we talked a bit in details about biosensors. A biosensor is an analytical device used for detection of chemical substance that combines a biological component with a physiochemical detector. The sensitive biological element could be tissue, microorganism, organelle, cell receptor, enzyme, antibody, nucleic acid, etc. Is a biologically derived material or biomimetic component that interacts, binds, or recognizes the analyte under study, right? So this is your biosensors. And of course, if you look at a biosensor, it will have a biological recognition element. This is the element that will first interact with your analyte or a biological material. That would then result in a signal which will be converted by a transducer into a quantitable commodity. Then this will be converted into an electrical signal to the amplifier and finally to the monitor, where it will be displayed in terms of what is the measure that is there. So the first interface of a biosensor is between the biological recognition element and the actual biological molecule that you have. The transducer converts molecular biological signal. What is the function of a transducer? the transducer converts the molecular biological signal into a digital or electrical signal proportional to the concentration or physiochemical spatial properties of the analyte, and can be amplified, quantified, displayed, and analyzed through the processor. So all these are processors, and then you have a transducer, right? Then, of course, there is one more diagram I showed you. So here you have a biologically sensitive element, you have a biological material. The first thing that happens when a biosensor is put to use is the interaction between the biological material and the biological response element or the biologically sensitive element. Once this is done, the transducer will detect the interaction, quantify it, and then convert it into an electrical signal. And then finally, you have a display. You could have a reference. So the first is the biological element interaction with the biological sensitive element. That would give you a biological response, which will be converted by the transducer into an electrical response, and then you have the measurement. So then, of course, biosensors, if you do not talk in very strict terms and you're not talking in terms of nanotechnology, then your normal digital thermometer is also a biosensor. So this is also a biosensor. Then in these days, we are living in COVID conditions, so to measure your oxygen content, or a distance thermometer is also a biosensor. And then, of course, a pregnancy kit is also a biosensor. And what we want now is a kit, something similar to pregnancy kit, where you can do a COVID-19 testing on a paper strip, and you can see a pattern that is there. This pattern would confirm whether the individual has COVID, does not have COVID. Of course, right now we are using real-time PCR to test individuals. Okay. So what is a nanobiosensor advantage? They are highly specific. Biological response element and the biological analyte, that interaction that happens is highly specific. If there is a bit of a change, you will not see a interaction, right? So there will be no reading. So in that sense, they are highly specific. Then they are highly sensitive, in the sense, even if the amount of biological material is less, you would still be able to measure and quantify it. Then, of course, it is miniaturization, which is important. When you use nanotechnology or nanobiosensor, you are ideally wanting to bring down the size of the biosensor or the machine that you are wanting to make. And why is that important? Because you want to use the machine at the point of care. Point of care is where the patient is, not where the hospital is. Right now, what we do is we have big machines, we have big X-ray machines and so on and so forth, MRI, this, that. So the patient has to go to the hospital and then get tested. With a setting in a country like India, where the hospital density is low, what you want ideally is that the machine should go to the patient, and that can only be done if the machine is small in size, requires less energy, less battery, and has greater portability, right? So biosensors have these advantages. They are miniature, so they are very small in size, therefore they can be used in point of care. They have greater portability, therefore they can be used as point of care device. Then higher sensitivity and higher specificity we have talked of, right? So here you could be asked, what is a nano device that is used for sequencing? So that will be your MinION. There are other seed devices also, but they are not at the nanoscale. They do not use a nanopore. So you could have something like an Illumina machine. You could have something like an ion torrent. You could have something like, let's say, Sanger sequencer or other sequencers, but they are not portable. They are big machines. They cannot be moved around. In contrast, this is a nanopore-based sequencer. It is called MinION, right? So MinION can be held in hand. It is more like a pen drive, and it can be directly used at the point of care for sequencing. Also remember why this is important, because now sequencing is fast becoming the first layer of diagnostics. We are anything and everything that you have, we are moving into an era of genomics. We are moving into an era where the patient, when he goes to the doctor, he would have to have his sequence in hand. And likewise, if there is an infection, you would want to sequence microorganisms in his blood to detect what is the type of infection that is there, right? So therefore, now sequencing is becoming what is known as the new microscope. So everything goes through sequencing now. So therefore, such machines are becoming heavily important, and we talked of this in more details. I'll show you a bit more now. So if you see, this is a Sanger sequencer, this is a Pyro sequencer, this is a Illumina machine, this is ion torrent. These are all big machines, at least the size of a large TV, if you see. So they cannot be taken to the patient. The patient has to come to them. The sample has to come to this machine. In this case, because this is using a nanotechnology where, and I'll talk of nanotechnology it is using, you can directly take it to the patient. You can just chip it to their laptop and this would work. So therefore now nanotechnology or nanobiosensors are becoming very important because they have this capacity of being point of care machines, right? Or miniaturization and high portability, and of course high sensitivity and high specificity. Then of course we talked of nanopore. I told you basically that you could have a natural protein that can work as a nanopore. So here you have alpha hemolysin. If you see, this is the tunnel. This is the diameter of this tunnel is in nano dimensions, and therefore you can use this as a nanopore. The principle is simple. What you do is, this nanopore is put into an electric field. Because of the electric field, there is a certain basal current that flows through the nanopore. And then when you obstruct it with some molecule that gets in, this current is characteristically changed. There is fluctuation in the current, and that is indicative of what type of molecule is possibly going through. And luckily for us, for adenine, thymine, cytosine, and guanine, the changes in current are distinct and of different types. And therefore, when you notice the change in current, you know which type of nucleotide is passing through the nanopore as of now. So that is what is allowing us to sequence without having to synthesize a complementary strand. All of the techniques that are there before this, they are basically using DNA polymerase. They synthesize a complementary strand, and based on what is getting added on the complementary strand, you get the sequence. Here in this case, you're not synthesizing a complementary strand. You are simply passing one strand of DNA through this and then reading what is the nucleotide that is passing through the nanopore, because each of these 4 nucleotides will produce a specific characteristic change. So remember, you want a single stranded DNA to pass through, so you have to use helicase here. So you'll use helicase instead of DNA polymerase, which is used in all other sequencing techniques. Here you would use helicase, right? Helicase is the one that unwinds your DNA. So one strand will now open up, and that will be made to pass through the nanopore. And based on what is the fluctuation that you get here, you'll get your sequence, right? So what is sequencing? Sequencing means to determine the order of nucleotides, A, T, G, C in a given DNA molecule. Genome, one full complement of genetic material in an organism, is called the genome. Why sequence genomes? We want to understand the basic composition between human and other genomes. We also want to identify the variations that are there. And more specifically for biomedical applications, we want to identify what variations associate with disease.